So to control microbial growth, we've got physical methods and then also a bunch of chemicals we can throw at them. And in this lecture, we're going to take a look at the physical control methods. So we'll go through all of these things on the left. We're going to start with heat, and we've got moist heat. So that includes boiling and autoclaves. And then we'll take a look at pasteurization and dry heat. And before we get into those, we've got two definitions here that are actually important if you're manufacturing food, especially canned goods, because we worry about, with canned food, botulism. And so we want to make sure that we kill off the Clostridium botulinum endospores and make sure that they're not in the canned food. So we've got two terms here. We've got the thermal death point, and this is just defined as the lowest temperature at which a specific bacteria will be killed in 10 minutes. So if you're looking at Clostridium botulinum, for example, what would be the lowest temperature that you could have that food at, that bacterium will be killed at in 10 minutes? And then the next definition, the thermal death time, is the minimum amount of time to kill a specific bacteria at a given temperature. So you just pick a particular temperature and then you figure out, well, how long is it going to take to kill Clostridium botulinum, for example, at that particular temperature? So if you have a hotter temperature, it'll take less time to kill the Clostridium botulinum, but if you have a lower temperature, it'll take a longer period of time to kill the Clostridium botulinum. So that's the thermal death time. All right, let's take a look at moist heat. This is boiling water, and how boiling water is going to kill microorganisms is by denaturing their proteins. So if you think about like cooking food, like maybe you're cooking an egg, the egg white will change from clear to white, and that's because you end up denaturing the proteins in the egg white because of the heat. And the same thing is going to happen when you boil the water, and if there's any microorganisms in there, you denature their proteins, they unfold, they don't work and do their job, and that kills the microorganisms. So boiling water will kill a lot of microorganisms, but it actually does not sterilize things. So that's really important to remember. So kind of our gold standard that we use to say if things are sterilized or not is if the bacterial endospores are killed. So boiling water does not kill bacterial endospores. So in order to kill bacterial endospores, you'd have to boil the water for 13 hours. And most people are not going to boil water for that long. So that's just not practical. But boiling water for 10 minutes, like maybe you're out camping, that will kill a lot of the other microorganisms and make it fairly safe to drink. But it's not going to kill the bacterial endospores. So it won't be sterile. Okay, but another way we could use heat, moist heat, is we can use an autoclave. And so what we're going to do here is we're going to put steam under pressure, and that'll allow the steam to reach a much higher temperature. So the steam will reach a temperature of 121 degrees Celsius. And remember that water boils at 100 degrees Celsius. So we're going to reach a much higher temperature. And we've got the steam under 15 pounds of pressure, and we need it to be at that temperature and that pressure for 15 minutes to kill off the bacterial endospores. And then that means that we've killed off all the other microorganisms, and whatever it is we've stuck in the autoclave will now be sterile. Okay, so When you run an autoclave though, the cycle is not going to take you 15 minutes because it takes time to heat up, and then you've got to be at that temperature and pressure for 15 minutes, and then you've got to let off the pressure and cool down before you can actually open the autoclave. So it's not a 15-minute cycle, so it does take a little bit longer than that. The important thing about the autoclave, you have to be careful with plastics. Because you are reaching such a high temperature, check before you put any plastics in the autoclave, because a lot of plastics will melt and then they make a huge mess. So there are a few plastics you can autoclave, but most of them you can't. So check before you put anything plastic in the autoclave. All right, so let's just take a look at some pictures of an autoclave. So here's what an autoclave looks like. They come in different sizes. This is a small one that you would find like in a doctor, dentist, vet office. So they just fit on the countertop. And if you're working in a hospital, they have these giant walk-in autoclaves. So the packs will fit inside the autoclave, and then steam will be pumped in, or some of them will just have water that will fill in the bottom. And then you shut the door. They heat up, and if you have water in the bottom, it will then turn into steam. And then the steam will penetrate the packs and sterilize whatever you've got inside the packs there. There is a locking mechanism on the door because this is put under high pressure, and if there wasn't a lock, it would just blow the door open and you'd lose the pressure. So, and then there's a venting mechanism, usually on the top somewhere, that will, when you're done with the cycle, it will let the steam out and the pressure, and then once that is all out, then you can open the door and get your packs out. Okay, we've got some quality control things here to make sure that our autoclave is working. So on the outside of the packs, we've got this masking tape or autoclave tape. And when you put it on the outside of the pack, it just looks like masking tape, but when it goes through the autoclave and the hot steam hits it, there's a black indicator strip that will show up, and that just indicates that it reached the certain temperature and the steam hit it. So when you pull your packs out, you should check the tape on the outside of the pack and make sure that it's turned black like that. And then on the inside of the pack, when you've wrapped them, you put an indicator strip inside the pack. And so it starts off white, and when the steam hits it, it will turn black. So when the packs get opened during surgery, you can check and make sure the indicator strip turned black to make sure that the steam got all the way into the inside of the pack as well. So we use both of these. So we've got an indicator on the outside of the pack and then a strip on the inside of the pack to make sure that the steam hit the outside of the pack and then also got on the inside as well. Some of the instruments we just wrap in a kind of a plastic pouch, and there's an indicator strip that is on the outside of the pouch here that will turn black to indicate that the steam hit it. And then this down here on the bottom right, this is a really good quality control to make sure that your autoclave is really working. These little tubes actually contained endospores inside of them. And so you would just put that in with your packs, run it through the autoclave, and then you would pull this out, and there's a little glass ampule in there, and so you just kind of bend the little tube there, and that snaps the glass, releases the culture media, and that'll mix with the endospores, and then you would incubate this in a little incubator for 2 days, and then you come back and check the color. You want it to stay purple, which means the endospores are dead. They didn't grow. But if the endospores were not killed by the autoclave and they hatched back into bacteria and they were growing, the culture media turns yellow, and that means your autoclave is not working very well and did not kill the endospores, and so your packs are not sterile. Okay, so a lot of the hospitals and clinics will also run these through the autoclaves, and this is kind of the gold standard to make sure that your autoclaves are really sterilizing things, because you actually run endospores through there to see if they're getting killed. So, okay, we also have another form of heat we can use. This is pasteurization, developed by Louis Pasteur. And what you're going to be doing is heating liquids at a temperature below boiling. So we would take like milk and other beverages, and they run them through tubes, and then they briefly heat them up. So like milk, they would heat it up to about 160 degrees Fahrenheit. Boiling is about 212 degrees Fahrenheit, so you're not going to boil the milk, but you're going to heat it up enough that you're going to kill the pathogens. So you're not going to get sick from the milk, but you're not going to kill all of the bacteria. So there's still going to be bacteria in the milk. And you do that for about 15 seconds. And then you'll cool it back down and then package it up and then sell it. And we know that there's still bacteria in the milk because your milk will eventually spoil and go bad because there's still bacteria in it. Okay, so it does not sterilize the milk, but it does kill off the pathogens that would make you sick, but doesn't get rid of everything. So that's pasteurization. Okay, and our last form of heat we can use is a dry heat. And this is an oven that's being shown here. And this oven is actually used in manufacturing. So this is not something you'd see being used in a hospital or a clinic. And it's going to require a much higher temperature and also a much longer time to sterilize things and kill all of the microorganisms. So you can see that you'd be reaching 170 degrees Celsius instead of 121 degrees Celsius like for an autoclave, and it would take 2 hours to kill everything. So we're going to be using this for things that would be damaged by moist heat like steam from the autoclave. So we'll use this to sterilize like glassware because the steam can actually etch and scratch the glassware and make it cloudy, and then powders because steam from the autoclave mixing with a powder would make a gooey mess. And then if you had to sterilize like some sort of an oil, the steam would not, the water and oil don't mix very well, so that wouldn't work very well either. So okay, and then in the lab, you guys have been using another form of dry heat, sterilizing your inoculating loops. So this would be flaming or incineration. So you just burn up the microorganisms. So you guys have been using that in lab. Okay, we can also use refrigeration and freezing. And most of our microorganisms that cause disease in people do not reproduce at refrigerator temperature because remember they're mesophiles, so they are going to reproduce best at human body temperature, and it's just too cold in the fridge. An exception to that rule is a bacteria that's pictured here on the bottom, Listeria. Sometimes you'll hear about Listeria on the news. Sometimes there's some recalls for foods. And Listeria is not a good bacteria to ingest, especially if you're pregnant, because this bacteria can cross the placenta and infect the fetus and actually cause a miscarriage. And then also if the person is elderly or immunocompromised, this bacteria can leave the GI tract and then make its way into the central nervous system and cause meningitis and can actually kill the person. So it can be a pretty serious infection. We tend to see Listeria in unpasteurized cheeses, other soft cheeses like brie, lunch meat, hot dogs. Sometimes you'll see recalls. There was a recent one for lettuce. It's also been on cantaloupe, ice cream. So there's a number of different foods that listeria has been found on. And then also freezing does not kill a lot of the microorganisms. So they just go dormant. They don't reproduce in the freezer, but they're still alive. And if you take the meat out of the freezer and thaw it, they get to a temperature where they can start reproducing again. And then if you eat that raw chicken, you'll get sick. Okay, so kind of keep that in mind. So usually freezing doesn't kill them. Okay, desiccation is where you're going to dry things out, and remember that microorganisms need water to grow and stay alive. So it depends on the microorganism. Things like endospores can survive being dried out, and some of the viruses can too. So there's a couple ways we can dry things out. So simple desiccation would be, for example, like dried fruits or beef jerky. Okay, and then the next one, lyophilization. That is the fancy term for freeze drying. So we can certainly freeze dry food, but we can also use this in medicine as well. So we can actually lyophilize vaccines, and some drugs will be lyophilized. And how you know that this has been done is that the vaccine, for example, well, if you look in the vial, it'll be as a powder, and you'll have to reconstitute it. So you'll have to add like some sterile water or sterile saline to it to mix it up before you give it to the person. So that has been lyophilized, so it's been freeze dried. Okay, so how freeze drying works is they will freeze it very quickly to a very low temperature and put the material under a vacuum and pretty much suck the water out of it and dehydrate it, and then it can stay preserved for long periods of time. So that's lyophilization. Okay, we can also reduce water activity and kind of dry things out by adding lots of salt to something. So thinking about preserving food, so for example, like beef jerky with lots of salt on it, or salting fish to preserve it, or if you add a lot of sugar to it. So that's going to create a hypertonic environment. And what's going to happen to the bacterial cell if we place it in a hypertonic environment? So hopefully you remember that if we put our cell into a hypertonic environment with lots of sugar or salt outside here, remember water wants to follow the solutes, and so water is going to leave the cell and the cell is going to shrink, and that's going to cause plasmolysis. So as the bacterial cell shrinks, the plasma membrane gets pulled away from the cell wall and it rips, and that kills the bacterial cell, and so it dies. Okay, we also have radiation, another way to kill microorganisms. So radiation is going to damage the DNA. So that's how it's going to work. And we have two different types of radiation. We have ionizing radiation, and this includes things like gamma rays, which comes from radioactive material, X-rays, electron beams, and these all have, if we look on our spectrum here, so like gamma rays, X-rays, these all have a very short wavelength, meaning they have lots of energy. And so they're going to be able to penetrate really deeply through things, like think about X-rays going through bones. So lots of energy, and they can knock electrons off things and make ions out of things. And by knocking electrons off things, they make free radicals, and that can end up damaging the DNA. The non-ionizing radiation is the UV or ultraviolet light. And this doesn't have as short of a wavelength, it's a longer wavelength. And so it's not going to have as much energy. So it's not going to be able to knock electrons off things, it's not going to make ions, and it's not going to be able to penetrate as deeply. Okay, so like UV light, thinking about being out in the sun, it causes skin cancer, but it's not going to cause skin cancer. I mean, it's not going to cause cancer any deeper in your body, okay, because it can't penetrate any deeper. All right, so let's take a look at this ionizing radiation. This is only used in manufacturing because it's too dangerous to be working with gamma rays, radioactive material or X-rays or energy beams in a clinic or a hospital to try to sterilize things. So they have in the manufacturing facility, they're going to have conveyor belt that will run whatever it is you're going to sterilize through this facility here, and this is all going to be encased to protect people from the radioactive material that's in here, or X-ray beams, or whatever you're using to radiate the surgery gloves, suture material, whatever you're running through here, so people are not in there at all. And this does not require any heat, so you can use this for things that are sensitive to heat, so that's really good as well. If you look on packages, if you're working in a hospital or clinic and you see sterile with an R, you know that it's been sterilized with this ionizing radiation, and that's how it's been done. So some of the injectable drugs that we use, because you're not heating it up, they can be sterilized this way, because if you heat the drug up, you could destroy it. You could denature the drug. Suture material, surgery gloves, plastics. You can even use ionizing radiation on food as well. So there's a lot of things you can use here. Okay, non-ionizing radiation will be the UV light, and UV light will damage the DNA by causing these thymine dimers. So what happens is anywhere you've got two T's adjacent to each other in the DNA, you're going to form covalent bonds between the T's where there shouldn't be any. And so you end up with a bump in the DNA from that covalent bond. And that's going to interfere with DNA replication and transcription, and so that's going to cause some problems. The UV light is not going to penetrate as deeply as the ionizing radiation, so you can only use this for surfaces. So they'll use this to sterilize like rooms, like a surgery room or a lab, and then they also will use this to sterilize like water. So some swimming pools or drinking water supplies, you can actually sterilize water with this. So they will use that. Okay, filtration. We can sterilize liquids or gases, and we're not using any heat by doing this. So let's take a look at filtering liquids. And so if you've got an injectable drug, maybe an antibiotic or something, and you want to sterilize that, so this will be done by the manufacturer. They will just run it through a filter with really small holes that microorganisms can't fit through, and then the drug coming out the other side will be sterile. And so you haven't had to use any heat at all, and so that will make the drug sterile that you inject into the patient. And you haven't ruined the drug by heating it up, denatured it, or anything like that. So filtration is going to be really good to use to if you need to sterilize drugs. You can either use filtration, or you could use the ionizing radiation. Okay, so that would be good to know. And especially if you're going into pharmacy, you'll be talking about that as well. Okay. All right, and then unfortunately, we've all learned about N95 masks and probably have been wearing them because of COVID. So these are very fitted, very tight, making sure that the air does not go around the mask because you want the air going through the mask. And they're called N95 masks because they filter out 95% of the particles in the air that you're breathing in. So they're pretty good at keeping viruses and things out, so you don't breathe those in. And then we also have HEPA filters. So these are used in hospital air filtration systems, and also if you're working in a lab in the biological safety hoods, they'll be used there too. So they've got really small holes in the HEPA filters that will trap the viruses or other microorganisms, and then they don't get, sprayed out again by the air filtration system. Okay, and then this is just from your book. We didn't talk about all of the physical methods to control microorganisms, but we've talked about a lot of these. So